and I will go ahead and kick us off here. So I just wanted to say hello and welcome to our webinar that's focused on accelerating population health research with integrative omics and exploring metabolomics data in large cohorts. So I'm your host today. I'm Dr. Micaiah Ward, and I'm a field metabolomics scientist at Metabolon, and I'll be running us through today's webinar. So we do have two amazing speakers lined up for today. Dr. Daniel Panyard and Dr. Kechris, and they'll be sharing their research with us. And I know I am very excited to hear about what they have to share. So before we officially get started, I just wanted to give a very brief introduction to Metabolon. So Metabolon is the global leader of metabolomic services. Over the last 20 years, we've developed unrivaled capabilities in metabolomic profiling, really setting the standard of metabolite coverage and data quality. So from untargeted metabolomics all the way through more quantitative targeted panels, and all of these are designed to really focus on your research needs. So Metabolon is very committed to advancing the field of metabolomics and biological understanding, particularly in human health and disease. So why would you choose to use metabolomics for your research? Well, metabolomics provides a snapshot of the entire physiology of a biological system. So most of us are familiar with the more traditional omics approaches that focus on the central dogma of biology, namely genomics, transcriptomics, and proteomics. So we have learned and we continue to learn very valuable information from these data, but they each only capture a single layer of the elaborate and integrative biological system. So even when combined, these data are still missing essential components that contribute to the biological complexity. So metabolomics, on the other hand, incorporates the effect of drugs, diet, lifestyle, and microbiome interactions, and collectively, this represents what we call the definitive molecular phenotype. So we like to think of metabolomics as the missing link in the central dogma, and that it's highly complementary to these other larger omics data sets, and when integrated, provides a comprehensive picture of the molecular landscape. This ultimately broadens our understanding of complex mechanisms and biological function. So that's really what we're going to be discussing today, specifically in the realm of population health data sets from large cohorts. So today we'll learn how to leverage metabolomics data in multi-omics research to magnify our understanding of human disease and identify actionable outcomes from these data. So like I said, we have two amazing speakers lined up for today, Dr. Daniel Panyard and Dr. Kechris, and they'll share their stories of how they integrated metabolomics data into their larger omics data sets and how this led them to a more comprehensive understanding in both Alzheimer's disease and chronic pulmonary obstructive disease. So first, I'd like to introduce you to Dr. Daniel or Danny Panyard. He's a postdoctoral research fellow in the Stanford Department of Genetics and a research associate with the Department of Veterans Affairs. So his research in molecular epidemiology and precision health focuses on characterizing large-scale genomic, proteomic, and metabolomic data in diverse cohorts and integrating these data with clinical data to uncover the biological systems underlying conditions like Alzheimer's disease. He primarily works with population cohort data like those from the Million Veteran Program and also the Alzheimer's Disease Research Centers. So his recent work includes an integrative proteomic and metabolomic analysis of Alzheimer's, a study of microsampling based on multi-omics for monitoring longitudinal and personal molecular changes, and also a comprehensive review of the metabolomics of aging. So today he'll be sharing his strategies for leveraging untargeted metabolomics in population cohort studies. So from here, I will let Dr. Panyard take it away.
Great. So I'm excited to talk to you all today about integrative multi-omics, especially with metabolomic data. So I'll go ahead and dive right in. We don't have a ton of time, so I will be going over a pair of studies that I think are going to be very useful to the audience here. I won't be going into great detail, but the links to these publications will be in the resources tab of the webinar. So if you have any questions or interested in some of the specifics of what we did, you should be able to find that in the papers there. So a brief bit about myself. As Dr. Ward mentioned, I'm a molecular epidemiologist. I work with genomic, proteomic, and metabolomic data. And in my recent work, I've been really interested in applying these approaches to studying a few different conditions, such as Alzheimer's disease, which I did with the Wisconsin Alzheimer's Disease Research Center, and also conditions of aging and precision health, which has been my focus here at Stanford. And then most recently, I've been doing a lot more general population omics work with the VA and the Million Veteran Program. My goal for today's talk really is to address a situation that I think a lot of folks find themselves in nowadays with large-scale population studies. And that situation is this. Imagine you have a disease of interest, and we really want to understand why some people get that particular disease and some people do not. Now, the tools for a long time included gathering data from the clinical side, would that be just diagnosis information, prognosis, clinical tests and assays, as well as survey information, gathering information about a patient's lifestyle or other risk factors. But in recent years, we've had the ability to start adding a lot more detailed molecular measurements to these cohorts. So genetics really being kind of on the first big data sets we've been able to add in. As the technology matured, the cost of these technologies became feasible for us to add for cohort studies on the order of hundreds, thousands, or even beyond that now. So genomics were added in, and then later on in recent decades, we've started adding in proteomics and lipidomics, mRNA sequencing data to get transcriptomic measurements. And of course, the focus of today's talk, adding in metabolomic information. Now, if you have an ongoing cohort study and you add in some of these omics data sets, particularly metabolomics, of course, one of the first things we might want to do is to just look at the standard associations of those metabolites with the main outcomes of our disease. So we might look at each individual metabolite and ask, is that metabolite associated with people who are getting our disease or with the biomarker that tracks that disease happening? But the question is, after you do those initial low-hanging fruit sort of analysis, the question is, what now? What else can we do with these data? And when you think about all the information that we have, especially with the high dimensional data sets across these different omic layers, there's a lot of interest in how do we combine this information together to drive new insight into the diseases we're trying to study. So that's my goal today, to share a little bit about some approaches I've used in my research, combining these different data sets together. And specifically, we're going to talk about two different case studies. One that discusses combining metabolomics with genomic information, and the second study will be a combination of metabolomics with proteomic data. So let's go ahead and dive into the first one. That's going to be integrating genomic and metabolomic information. So this is a study that came out a couple of years ago. It's actually my dissertation work. And it's all about understanding the genetic regulation of CSF metabolites and understanding how that relates to disease. So it's an open access paper, so you should be able to find that online in its entirety. So a little bit about the study background so you understand the motivation of what we were doing. And really the concept was fairly straightforward. We know that brain metabolism can be affected in neurological disease, especially Alzheimer's disease, which was kind of my group's primary interest. And we know that if you want to study the changing brain metabolism, the cerebrospinal fluid is going to be the sample type of most interest. And that's because that's the fluid that's in direct contact with the brain and the spinal cord. But the challenge was that, especially at the time, there just wasn't that much CSF metabolite data that was out there. I mean, there was some, but compared to blood metabolomics, there's just far fewer CSF metabolite resources. And so the question that we wanted to ask was how the genome of the CSF metabolome and neurological diseases were related, and ways we might be able to develop to help us study those relationships in more detail. So the data sets that we worked with in this project came from the Wisconsin Alzheimer's Disease Research Center. And in particular, two different cohorts, the Wisconsin ADRC's main cohort, and then a study called the Wisconsin Registry for Alzheimer's Prevention, or the RAP study. Overall sample size for this particular kind of sub-study in this group, we had about 300 total samples. You can see the main demographics listed there. And on this particular cohort that we were studying, specifically, these numbers refer to those individuals on whom we had full genomic information from genome-wide genotyping. So we're talking after imputation, over 7 million SNPs in each cohort. And then we had also generated CSF metabolomics data using Metabolon's platform, that gave us information for 338 different metabolites. And you can see we captured a broad range of biology in this data set. You can see the molecular classes of the metabolites listed there, covering things from amino acids to lipids, xenobiotics, peptides, and so forth. So with these data, we were interested in developing a new method to combine genetics and metabolomics to study neurological disease. And we call this overall approach an MWAS, or metabolome-wide association study. And the name is really a play off of the TWAS methodology. If you're familiar with that approach, it uses genetics to impute gene expression and then study the relationship of gene expression with disease. We're basically doing the same thing, but we're extending that method to metabolomics. If you're not familiar with TWAS, this is kind of the overall concept of what we're doing here. Imagine you got three main data sets of interest: genetics, metabolites, in our case in CSF, and then brain phenotypes. What you do is in one data set where you have genetic and metabolomic information, you build predictive models to let you predict CSF metabolite levels using genetic information. And then once you have those models built, you transport them to a second data set where you only have genetic and outcome information. In our case, different brain-related diseases and phenotypes. And in that data set, you use your models to predict the level of each CSF metabolite, and then you use those predicted metabolite levels to study the association of those metabolites with your brain phenotypes. So to set up and support this method, there were three stages of analysis we went through. The first was just understanding the relationship between genotypes and CSF metabolites, which in our case was conducting a GWAS, a genome-wide association study of each CSF metabolite in our data set, just to understand what the genetic architecture was. And then stage two, we built prediction models for each CSF metabolite using a couple of different regression modeling approaches. And then third, and finally, this is the payoff of the method, we use those predictive models to impute CSF metabolite levels in external GWAS summary statistic files, which allowed us to effectively study metabolite disease associations. And this is using a software package called Badgers. Okay, so again, we don't have a whole lot of time to go into the details. There's a lot covered in the papers. I'm going to focus just on the highlights insofar as that relates to our discussion today. So first of all, what does the genetic architecture of CSF metabolites look like? So this is a summary of the results of all the 338 different GWAS that I ran of CSF metabolite levels. What this shows is a stacked Manhattan plot. Each dot on this figure tells you the strength of association, the p-value, of each genetic variant we studied and its association with one of the CSF metabolites. Now, across the 338 metabolites that we studied, we had significant associations after multiple testing for 16 of those metabolites. And so the stacked image shows each of those 16 significant loci here on the same figure. So you can see them all at once. One thing I want to point out here, if you remember, our sample size was about 300. Now, typically, if you're familiar with GWAS data sets, usually they number in the order of thousands, tens of thousands, even beyond that now. So note that we had a very small sample, but we had very strong association findings for a number of metabolites. And assuredly, we were missing some associations in here just due to a lack of power. I'm pointing this out because it's really interesting that when you study metabolite levels and you study their genetics, you often find that there can be very strong effect sizes, much stronger than you see for typical genetic disease associations in most GWAS that are out there. So these molecular level associations can really yield some insights even at lower sample sizes, which again makes it feasible for a lot of us in working with smaller population cohorts. Okay, so that's just one interesting thing to point out about kind of the metabolomics world here. Okay, but if we zoom into these specific GWAS loci and we look at the specific genes wherein those variants are located that are associated with these metabolites, a few different themes emerge that I think are important to point out for those of us integrating these data sets together. The first is that if you look at the typical gene associated with metabolites level in a GWAS, what we found was that these genes often were for protein transporters for metabolites or enzymes using the associated metabolite as a substrate. So really what that means is something very interpretable biologically, which is that these proteins whose mutations tend to affect metabolite levels often are closely connected to those metabolites in a biological pathway. So the interpretation often is fairly straightforward for a lot of these loci, because it's pretty clear to understand why a mutation would be affecting that metabolites level. What was interesting is that when we did the study too, we found a number of novel associations with metabolites that hadn't been reported before. And again, our study was smaller than a lot of other studies that were out there. In a few cases, this was because we were studying metabolites that just hadn't made into the literature yet. But in a few other cases, the gold rows here, betaine, oxalate, and guanosine, we found associations that for these metabolites, even though these metabolites had been studied before in blood, we found associations for loci that hadn't been reported before, and presumably might be due to unique genetic regulation in the cerebrospinal fluid. All this to say that context really matters. Even when you're doing a GWAS, even though genetics are more or less, kind of consistent throughout the life course, the context still matters when you study these associations. If you have a unique population or unique disease that you're looking into, unique sample type, you might still find novel associations. So I think it's still worth it to ask these questions with your data sets when you have genetic information. You still might find something that hasn't kind of been reported in the past. Okay, so on to stage 2. We've got a general sense of what the architecture of metabolites look like. Now, can we leverage this genetic data to build predictive models for these metabolites? Okay, and this time I'm going to skip over a lot of the details here just for the sake of time, but the interesting things about building these sorts of models is that the approach is a little bit less straightforward than it is for other kinds of molecular data. With metabolites, if you're trying to predict them, what's tough is that we don't really have a straightforward genetic architecture for all metabolites that would work. If you're predicting protein levels or gene expression, it's reasonable to look at genetic variants located within or near the gene for that protein or that transcript. Metabolites don't map to the genome. So when we're building these models, we have to allow for SNPs or genetic variants from across the genome. And so that just adds a little bit more computational challenge to it. You also have to allow for varying degrees of sparsity. Some metabolites only need a couple of genetic variants. Some are more polygenic in their regulation. So you have to be flexible in what you use. In our case, we used a combination of polygenic risk score or kind of general linear models, and then also elastic net regression. We ended up doing a lot of cross-validation model selection, tried a lot of different types of models to see which ones work best for different metabolites. I'll skip to kind of the final results of this process, which are summarized here. Each metabolite is a different vertical bar, and the height of the bar tells you how strongly the predictive models worked for that metabolite. And as you can see, if you look at the y-axis here, we're looking at R squared, or proportion of variance captured, for most metabolites around 5%, in that range, about 2 to 5%. So we're capturing just a modest amount of variance in these metabolites in most cases, but there are a few notable exceptions on the far left-hand side. There's a handful of metabolites which are very strongly predicted by even just a couple of genetic variants. So again, just reiterating this theme that when you study genetics and metabolites, you can often find these very strong associations with metabolite levels in a few cases. At any rate, we built these predictive models, and the goal then is for, I think, about a third of these metabolites that had a fairly good amount of variance captured. We then wanted to use this MWAS approach and understand how these metabolites are related to brain phenotypes. Again, the excitement here is that we're studying metabolites that often hadn't been studied in particular disease contexts before by leveraging these genetic models. So we're extending our insights from one data set to data sets where people just haven't been able to collect individual level metabolomic data yet. So the approach here, as I mentioned before, is to test these metabolite phenotype associations. We're using the Badger software, again, similar to the TWAS methodology. But what's great about this approach is that it allows us to simply use GWAS summary statistics in combination with our genetic prediction models. So we don't even need individual level data to do so. Now, the trick is, because we're predicting associations for metabolites that often hadn't been directly studied in disease yet, how do we validate our findings when the data haven't been collected yet? So we looked to a variety of experimental literature to see if our predictions were feasible. And then we also tried using two-sample Mendelian randomization as kind of a orthogonal but similar approach to what we were doing here. We ended up finding 19 significant metabolite disease predictions. A lot of these ended up having some evidence in the literature. We discussed these in detail in the paper, but for now, I'll just mention kind of 3 examples here. All 3 of these metabolites, alpha-tocopherol, N6-methylysine, and N-delta-acetyl-ornithine, we predicted to be associated with schizophrenia. And it turns out in the literature, you can see some examples from experimental models in the past, which indicated these molecules or implicated them as possibly being related. And actually, just a couple of weeks ago, this MWAS paper that we wrote was cited in a study of epilepsy. It turned out in a lot of our prediction models, N-delta-acetyl-ornithine popped up repeatedly as a metabolite we were finding to be associated with a number of different neurological phenotypes. And interestingly, again, the study that just came out, we're using an experimental model of epilepsy. And what did they find? Well, it turns out N-acetyl-ornithine was related in their actual direct measurements too. So they cited this paper as kind of being one of those early indicators of this metabolomic association. So just exciting to see, again, this overall approach bearing fruit even now in 2023 and 2024. Okay, so all this is just kind of one way to leverage genetic data and kind of what the genetic metabolite association landscape looks like. So now in the remaining time that we have, I'm going to talk about a different situation where you have proteomics and metabolomics information together in a cohort study. Again, we'll be talking about Alzheimer's disease. This is a multi-omics study that I conducted over a course of several years. The paper just came out last year, talking about, again, kind of proteomic metabolomic combination in AD. The data set came from the same fundamental cohorts that I mentioned before from Wisconsin. In this case, for the proteomics data set, we had 137 participants with CSF data or CSF samples available, on which we measured the levels of 915 different proteins in those CSF samples. We used a mass spec approach to do so. Now, what was great is that when we added these proteomics data, it was added in the context of these samples on which we already had pre-existing Alzheimer's biomarker information for 9 different biomarkers. We had the metabolomics data that I mentioned before. So here we had 390 different metabolites. We also had full genotyping information for all of these individuals. And all the CSF information were available on the exact same CSF samples for these participants. So we have this concordance of measurements here that gives a really rich data set. So even though we have just 137 participants, we have a lot of different measurements available to dive into. Now, the bulk of this paper was all about proteomics for the most part. And so most of the analyses I'm going to be skipping here, but the paper has a lot of other stuff if you're interested in this broader story. The proteomic associations with the biomarkers is summarized here. You can see each one of the 9, well, in this case, we only had 8 biomarkers with significant associations with proteins. But those associations are shown here as a network. You can see general clusters of those associations based off the biomarkers that proteins were associated with. But that's not the focus of our talk today. What emerged when I looked at the proteomics was a theme of a general pathway being altered or dysregulated, kind of an early Alzheimer's pathology. And that theme was carbon metabolism, glycolysis, gluconeogenesis. It was the protein enzymes of these central carbon pathways that were increasing, as it were, in these samples as kind of these preclinical stages developed for Alzheimer's. So our question was really, well, we see this general pathway association. Can we dive into more specifics of what's happening? Enter the metabolomics information. So remember, this is a new proteomics data set, but we have this metabolomic information already measured. And so using our data set from Metabolon, we were able to quickly use the annotations from the provided files to identify 10 different metabolites we had measured that were in related pathways. So metabolites known to be connected to glycolysis, gluconeogenesis, the TCA cycle. So this gave us a very straightforward and justified hypothesis to test. Are these metabolites also changing at the same time that these proteins are changing? So we did a quick metabolite association analysis in these samples. So we used the exact proteomic data set as our discovery, and then we had additional samples that didn't have proteomics, but that did have metabolomic information. So we used those as a replication data set to understand which of these 10 metabolites were associated with our primary markers of Alzheimer's development. And as it turns out, one of them did. It was succinylcarnitine. You can see in the box plots here that as we kind of progress through these preclinical stages of of Alzheimer's progression through its biomarkers. As amyloid and tau biomarkers become positive in this cohort, succinylcarnitine levels were increasing. And it wasn't just our cohort. Actually, I think a little bit later in the year that we posted the preprint of these results, another cohort investigating the metabolomics of AD also found a similar theme. They found altered acylcarnitines as one of their major findings. So great. So we were able to use our metabolomic information to refine our proteomic findings and really kind of dive into the pathway in a way we wouldn't be able to do if we only had the proteomic data set. Okay, so we're about out of time, so I'm going to take a moment just to wrap up with a few thoughts. And again, imagining that a lot of us are in this situation where we've got multiple omic data sets together. How do we combine them? What sorts of questions should we be asking? Well, when you have genetic data, consider asking what is the genetic architecture of the metabolites you've measured. And again, remember that even at smaller sample sizes, you can still often find stronger genetic associations. And the context can give you new insights if your particular population is different from what's been studied in the past. There might be some disease interaction going on where a particular pathway is only going to be active in a disease state, and maybe you can only find altered metabolite genetic association in that context. Or maybe your population just hasn't been studied very well before. So it's worth it to still ask these questions, even when there's larger cohorts out there. Second, you can use those genetic metabolite associations to then investigate other metabolite disease relationships, as we discussed today with the MWAS approach that I highlighted. And of course, and I didn't have time to get into this too, but there's other approaches to leveraging genetics that you can use to study metabolite disease associations. Mendelian randomization is probably the most popular approach of these. And should you satisfy those statistical assumptions, it actually kind of gets at the causal effects and causal directions too. So genetic data can really be a great way to augment metabolomics data. When you have proteomic data, what sorts of questions can be interesting to ask with your metabolites? Well, first of all, when you've got a particular proteomic pathway being altered, look into the metabolomics data. Align the annotation files, look to see if there's metabolites measured in those same pathways, and see if your metabolite findings coincide with what you see from the protein side. If it does, great, you've refined your signal a little bit further. If they don't align, that's still really interesting information. Why don't they align? What might be going on and what might explain that? Same thing too. Again, as we saw with the genetic findings and associations with metabolites, a lot of times you see that the proteins affecting metabolites tend to be enzymes or transporters for those metabolites. So if one of those proteins is dysregulated in your disease, are the downstream metabolites also being affected? If so, if not, either way, it's still going to be informative in some way. And then finally, too, this is a question I didn't have time to get into today, but in that second paper, we actually ended up building predictive models from different omic data sets individually to predict our main measurements of amyloid and tau protein as biomarkers for Alzheimer's disease. And so you can start to ask questions comparing these different omic data sets and understand to what extent are different types of data explaining or predicting your different outcomes of interest. And when you do so, you can start to understand a little bit better maybe what's going on a broader sense biologically with your disease and how these different data sets might be best leveraged. Okay, I'm going to end it there. I think we're about out of time. A lot of people to acknowledge on these particular papers. Again, it came from the Wisconsin ADRC and RAP studies. So the participants there who support these studies and the investigators and the staff and the students who've all worked on these, big thank you to all of them. Of course, the funding for these projects, including a training program is involved in a couple of different NIH studies and many more in the papers themselves. It takes a lot of folks to work together on large omics data sets like these. So definitely appreciate kind of the collaborative nature that we all had. Finally, too, I love talking about this stuff. I think it's a lot of fun. I think there's just so much to be done in the omics space. Always looking to collaborate more with other individuals. So if you're interested in what I talked about today, in the methods, or maybe in kind of the insights into Alzheimer's disease, feel free to reach out to me. I'm always happy to chat. So I think we're about out of time, so I'll stop there. But thank you all for your attention.
Wonderful. Thank you so much, Dr. Panyard. That was very, very insightful. I think we do have time for one quick question, so I can go ahead and ask that while I switch over the slide deck here. So the question is, how do you establish the cause and effect in this type of study? Metabolites might be changing as a result of disease, but not directly involved in disease etiology.
I love that question because it's one I ask all the time too. And that is one of the big challenges. So genetics set up birth for the most part, right? Metabolites, proteomics, other things like that, dynamic, highly dynamic. And as we saw with the GWAS associations and the predictive models, how much variance are we capturing just with genetics? Again, we might have been underpowered, but still we're talking very modest amounts. Lots of influences on a lot of metabolites that are non-genetic. So how do you get at causal relationships? Well, again, statistically, there is this idea you can use Mendelian randomization, aka it's just an instrumental variable analysis at heart. And if you can find predictors of metabolites that are unique and you satisfy some other assumptions, theoretically, you get at those causal relationships methodologically. Another way to get at the question that I think might help in cases where it's a little bit harder to find those, to satisfy those assumptions, which I think are a little bit harder in practice than we might think. Longitudinal data, I think, is going to be really useful for this to an extent. It's not going to be perfect, but if you can capture data across the span of disease from before it begins through when it does, that longitudinality, not only does it just let you see the trajectory of how things change, it also gives you, if you're measuring the same person multiple times, in some sense, you've got built-in controls. You compare one person to themselves over time. So that helps reduce some confounding. It's not perfect. Of course, other things change in life that might be happening. So of course, there's no easy answer. It's complicated. But that's the challenge I think we're all going to be working with as these data become more prevalent.
Yeah, perfect. I really like the inclusion of that longitudinal data aspect, because I think it's important with these big data sets for us all to remember that, especially with transcriptomics, proteomics, and metabolomics, we are really just capturing that snapshot. And so it's so highly dependent on the time that you sample. So yeah, I think that's an excellent point. So I will switch the slides over now. We will move on to a little bit more about Metabolon, and then we will hear from Dr. Kechris. So in addition to the fascinating work that you're hearing about today, metabolomics has applications across a broad area of research fields and really helps in overcoming traditional challenges of translational research. So in both basic and applied research areas, metabolomics offers solutions in understanding biology, the prediction of disease outcomes, enabling patient stratification in clinical trials, adding insight to understanding functional genomics, and ultimately linking the genotype to the phenotype. But tackling these challenges and moving the needle in translational research really requires access to consistent, high-quality data and insights. So Metabolon approaches this challenge with multiple panel offerings that range from the wide-net discovery applications of our global discovery panel, all the way through to targeted panels and even in pinpointing single analytes. So these panels have been designed to answer very different research questions, as well as address different applications needs. So our global discovery panel is our approach to untargeted metabolomics, and this offers unrivaled coverage using our proprietary library of over 5,400 metabolites. So our global discovery panel offers relative quantitation, and it's ideal for getting the most data out of your biological samples without specific knowledge of what biochemicals may be present. So with this panel, we can capture changes related to a variety of factors, and that includes our genes, exposure, diet, therapeutic, treatment, disease, all while delivering the best possible representation of the phenotype at any given point in time. So we also recently launched some more focused discovery panels, and these are really tailored for researchers primarily interested in either biological stress, kidney, or liver function. And these panels offer a relative quantitation report of a subset of the relevant metabolites for those particular focuses. And they come with a suite of advanced data visualization tools that are specifically designed to enhance interpretability of these large data sets. So if we navigate over to the right-hand side, we have a few examples of our targeted panels and single analyte assays. These are designed to build on and complement our discovery work. So they offer absolute quantification of metabolites, and most of these have previously been identified as important in a specific biological pathway. We offer 22 different targeted panels and assays, as well as the ability to entirely customize panels based on your specific research needs. So Metabolon truly sets the standard in metabolomics analysis, and we excel in coverage, identification, as well as data quality. So the main driver that enables us to offer the quality of results that we do is our extensive reference library, which includes both purchased and in-house synthesis of authentic chemical standards. These standards have been run repeatedly on our platform, and so this really enables us to characterize their exact molecular fingerprint. And our library covers over 5,400 metabolites from over 70 metabolic pathways, and this really allows us to offer the highest level of annotation confidence as well as depth of coverage. So this also allows us to provide insights into the multiple and diverse complex interactions that really contribute to that complete biological system. So what does working with us look like? What is it like having Metabolon as a partner in your research? So we really are with you every step of the way, where we provide guidance to power your study for success, all the way through to analysis and data visualization. So we really like to work with our investigators very early in the process to ensure that the study design and sample handling protocols will deliver the best possible results to answer your research questions. Once the samples arrive in our lab, we perform extractions with unique SOPs, and these are based on the sample type, and we do this prior to analyzing on our platform. So because the matrices that we work with and the analytes we cover are extremely diverse, so they have different biological makeup, physical chemical properties, it really does represent a great challenge in being able to accurately and repeatedly detect the very broad range of metabolites that we cover. So to address this challenge and fully capture the biological and biochemical complexity, we use different and complementary data acquisition methods, and these are designed to maximize the metabolite detection and coverage. So once we complete this step, the data is then matched against our reference library, and this turns tens of thousands of ion features into identified metabolites that meet our rigorous and stringent QA/QC requirements. And finally, the last step is matching our results to our reference library. And and so we really are able to identify metabolites to match biochemical pathways, and we can take you beyond the data table and elevate meaningful biological insights and ultimately help you narrate a comprehensive and credible story for your research discoveries. So as alluded to on the previous slide, mass spec data is very rich in noise. It also has lots of artifacts and redundant signals, and this may or may not be important or meaningful to your study. So how do we extract this biologically meaningful data and significant metabolites from extremely noisy data sets? So at Metabolon, we use what we call a chemocentric approach to address this challenge. So with each project, we generate approximately 150,000 ion features, but these 150,000 ion features do not necessarily translate to 150,000 metabolites in your sample. So we then take these features and search them against our annotated reference library, and these are, again, the pure authentic standards that we can measure against retention time, mass, and fragmentation patterns for each of the standards in our library. So our library contains a signature of the ionization products of every compound as well. And this is what really allows us to filter through and remove the noise and enable appropriate and accurate identification of those metabolites. It has the added benefit of reducing discovery, of false discovery, which is a huge issue, as we all know, and a common pitfall in these big data sets that we see in omics fields. So finally, after the reduction of the step to remove noise and extraneous artifacts from your data sets, we're then ready to move forward with a confident list of identified metabolites, and that these we can then further interrogate for biological meaning. So for our discovery platforms, we provide client data tables in multiple formats, and this allows researchers to directly import their data in their favorite analysis tools such as R or Python. And then for the targeted panels, we provide more quantitative data of the specific set of metabolites of interest, and this is immediately usable for statistical analysis and biomarker validation. Of course, our services do not stop at data generation. So beyond our client data tables, we offer statistical analyses and visualization tools that allow researchers to, again, immediately interpret key results. So we saw a lot of this with Danny's work. And so I think that's really important to highlight that the data that we provide back to you is really immediately interpretable. And even if you have no prior experience in metabolomics, this is really, really useful. So from here, we also offer in-depth and fully referenced interpretive reports. And these are written by our in-house discovery and translational sciences team, which is a team of over 40 PhDs of diverse biological, biochemical backgrounds. And they're really able to take full advantage of our wealth of institutional knowledge and guide you through the key results to provide actionable insights. And so as we proceed to our next speaker today, I really would like to invite you to ask yourself, what can Metabolon reveal for you? And so with that, I would like to introduce our second speaker today, Dr. Kechris, who is a professor of biostatistics and informatics, as well as the associate director of data science at the Center for Innovative Design and Analysis at the Colorado School of Public Health at the University of Colorado. So she received her undergraduate degree in applied mathematics from the University of California, Los Angeles, her PhD in statistics from the University of California, Berkeley, and before her appointment at the University of Colorado, she also completed a postdoctoral fellowship at the University of California, San Francisco in computational biology. So Dr. Kechris's research is focused on the development and application of statistical and computational methods for analyzing omics data sets through all stages of the data lifecycle. So this includes processing, storage, analysis, modeling, and visualization. So specific areas that she's focused on include analyzing transcription factor binding and microRNA expression data to study regulation. She's also examined the genetic and epigenetic factors controlling gene expression, exploring the metabolome, and integrating multi-omics data. So she collaborates with investigators in studying COPD, substance use disorders, and using animal models, and also in early life determinants of diabetes and obesity in children. So we're very excited to welcome Dr. Kechris and to hear about her work in multi-omics and network approaches for studying chronic obstructive pulmonary disease.
Thank you very much to the Metabolon team for organizing this series and for inviting me to speak today. As mentioned, I will talk about some of my work using integrative omics and network approaches, in particular with chronic obstructive pulmonary disease. So as I introduced already, I'm a statistician by training, and so I work on the development of methods for high-dimensional data analysis, and I've worked across the omics spectrum. Today, I'll be talking more about metabolomics and the integration of different omics with metabolomics. And all this work has been very much motivated by collaborations with investigators here on campus and beyond. And today I'll be talking about more of my work with pulmonologists. And this work, in addition to the applications, a lot of my lab is focused on methods development and software and databases. And I'll provide the link and for some of the software at the end. So moving along, today's focus will be on chronic obstructive pulmonary disease, COPD. And here I just have some examples of the sort of main phenotypes. It's a very heterogeneous disease, although for the most part, a lot of the standard definitions are based on measuring airflow obstruction using spirometry. And I have some of the variables that are measured, forced expiratory volume in 1 second and forced vital capacity. And these could be also grouped into categorical values for severity of COPD, from 0 to no to mild, all the way to 4, which is severe. But as I mentioned, this is a very heterogeneous disease, so we can also look at emphysema, which is another subtype of the disease, through CT scans, but also chronic bronchitis, these are questions from the clinical visits, and also whether an individual has frequent exacerbations which are acute episodes that require medical attention, either treatment or emergency room visits. So here are some statistics on COPD. This is a disease of aging, and so it's primarily in older smokers. Smoking is the primary exposure. However, not all smokers develop the disease. So there are a few modifying therapies. They tend to be sort of inhaler-based and using corticosteroids. And after cancer and heart disease and accidents, it's the number 4 cause of death, and has a large sort of impact in hospital days and also financial impact. So it's very complex, it's heterogeneous, and then we have all these comorbidities that are also associated with the aging process. So this next slide shows just some data showing the heterogeneity. So the x-axis is this spirometry-based airflow obstruction measurement, and sort of a line sort of indicating sort of severe or mild to no COPD with respect to spirometry. The y-axis is percent emphysema. This is based on the CT imaging. And then in color is a clinical sort of related question, whether a subject has chronic bronchitis. And you can see there's some association and some correlation, but however, there's lots of subjects that do not follow definitely all the extremes on these different phenotypes. So the heterogeneity is something that's of interest to understand, and that's where extensive clinical data and also all this omics data can help. So I work in the context of a large genetic epidemiology study called COPD Gene. It was started more than 15 years ago with several recruitment sites around the country. I was involved because Denver is one of the largest recruitment sites, and it is now in its 4th stage. So every 5 years there was a visit, phase 1, phase 2, and phase 3. Phase 3 has been completed, and now they're planning for phase 4. And it started out at baseline with more than 10,000 subjects. Yeah, I believe it's one of the largest in the world that selected for more extreme cases of COPD. So it was not just a population-based study. They selected for these extreme, more severe values. And they also selected to have a very good representation, relative representation of African Americans. So that's about 1/3 of the cohort. Some of the other demographics, like I said, this is a disease of aging. So these tend to be older individuals. At baseline, it was between 45 and 80 with a mean of 60. And because smoking was the primary exposure, at least definitely in the US, these were all individuals who had a history of smoking, at least 10 pack years, and either were current or former smokers. And there's a small set of a few hundred of non-smokers that were used as controls. So because this was a genetic epidemiology study as a design, there was extensive genotyping at baseline using SNP chips. But since then, COPD Gene, like some other large cohorts around the country, have become part of a larger consortium called TopMed that is funded by NIH and NHLBI. So we also have whole genome sequencing now through that effort. In addition to, they provided funding to also perform other omics profiling. So extensive genotyping, extensive clinical variables, hundreds of different variables, and all the imaging that's performed at every visit in these multiple time points. And actually, as mentioned in the previous Q&A, that's where we can use this longitudinal data to start to ask maybe more sort of looking at causation. So this is the overall sort of picture of the study. How was I involved? Well, initially, yes, it's very much focused on GWAS, but there have been so many different ancillary studies and offshoots of the project because of the rich data sources. So I've been involved with a variety of different omics here. I listed DNA methylation and transcript and proteins and metabolites that were measured typically on blood from these subjects. And we can use these different profiles for a variety of tasks to ask different scientific questions. And I'll go through each of these today. Some focus on biomarker discovery, thinking about mechanisms, and also molecular subtyping. So let's first start with the biomarker discovery. So as our group started to generate different omics data, we first started with a single omics analysis and just trying to find what are the proteins or metabolites or genes that are associated with some disease phenotype. Since this was in blood, we consider this to be biomarkers. And I just have examples of studies. We've published dozens of studies in this area, looking at COPD severity and progression and mortality, looking certain classes of biomarkers like cytokines and chemokines, looking at not only proteins, but also gene expression, and also other types of phenotypes like these exacerbations, history, and also linking with other cohorts. It is critical with this biomarker discovery to be able to replicate results in other cohorts. So almost all these studies have replication using another cohort. In this case, spiromics is one that we often work with. Then we've started to work more in metabolomics. This was a study that we actually had plasma and also another sample type called bronchoalveolar lavage fluid, where fluid is squirted into the lung and you can extract cells from the lining of the lung. So here we were able to compare the metabolomic profiles between these sample types. And finally, we were also linking these biomarker studies with genetics. Here's one biomarker that's been very well studied by many groups and called SRAGE. And it's been found that genetic ancestry in certain variants affects the association of the biomarker with COPD. So it's only a good biomarker in certain contexts and certain populations. So it started to lend itself to this more precision medicine approach. So these were all the single omics studies. As I mentioned, we started to get into more metabolomics. So I'll kind of mention some types of mechanisms that are important in COPD. Related to metabolic processes. And one is the role of sphingolipids, which is a class of lipids. And this has been well studied in COPD, especially two types, one called ceramide and one called sphingosine 1-phosphate, S1P, and which are in an equilibrium. And due to smoking and other exposures, this equilibrium can become imbalanced and then cause oxidative stress and inflammation, leading to emphysema. So a very well studied sort of metabolic process for COPD. However, only these two sort of types of single lipids were studied in great detail. So that's when we started to think about metabolomic studies, and we did both targeted and untargeted to look at this pathway in more detail. And that's what I have here is the pathway, sort of metabolic pathway, where ceramide and sphingosine 1P are here in red. But then we were able to fill in a lot of the gaps and look at a lot of the other sphingolipids. And you can, in the arrows, kind of denote whether those sphingolipids were up or down related to different COPD phenotypes. But then layering onto that, we had gene expression through a transcriptomic study on the same subject. So what I have here in sort of the italics are the enzymes that are related to different steps in this pathway. And we also saw similar sort of behaviors where some were over or underexpressed related to the phenotypes. So this was kind of the beginning of our approach to starting to think about multi-omics analyses, and using this particular pathway kind of motivated how do we study this, this pathway or others in more detail, and how do we identify new pathways. So that led to the thinking about multi-omics projects, and as we generated more data on the same subjects, we were able to kind of think about what questions can we ask. So here I have a sort of a conceptual figure here of where you can think about rows are subjects, and then columns are different omics profiles, whether they're genes or microRNAs or proteins and metabolites. And depending on the question, we may look at this data in different ways. Okay, so one question is thinking about mechanisms. So one approach that I'll talk about next is how did we identify multi-omics molecular networks? So you can think about it's a way of grouping or clustering these features that are show association, similar to kind of the work with the sphingolipid, but now in a just agnostic way. So I'll talk about that work, and then next I'll talk about kind of the flip side, thinking more about the subjects. Because of the heterogeneity of COPD, there's of interest of identifying new subtypes. Now, there's been a lot of work done with the clinical variables, but how can we use all these multi-omics profiles to define subtypes, and how do they complement the clinical subtyping? So I'll talk about that as well. So starting first with the network approach in studying mechanisms. So this was several years ago, and at the time, most methods for identifying networks tended to be single omics. They weren't quite designed yet for multi-omics. And they didn't necessarily consider phenotypes or traits or disease severity. They were just trying to find networks of features that were highly correlated. But we wanted to anchor that with one of the phenotypes because of the multidimensional sort of nature of COPD disease and phenotypes. And another aspect is more the informatics, is that these methods can be very sensitive to parameters. There's a lot of tuning parameters on the way, and you can get very different results. So it's just a conceptual figure here, and I have an example with microRNA and mRNA, but I'll come back to metabolomics. This was just a motivating example when we developed the method, but the idea is we have, for the same subjects, messenger RNA from transcriptomic study, microRNA from also non-coding, sort of transcriptomic study, and a trait of interest, and we want to find trait-specific networks that are anchored to a particular trait because of the, again, I said, the high-dimensional sort of traits associated with COPD. So a postdoc in the group at the time developed a method called sparse multiple canonical correlation network analysis. And this is sort of a conceptual figure. There's sort of it's a sort of a computational approach behind the scene and an objective function that's being fit. But the idea is, what is, say we have genes and microRNA, and to do a multivariate correlation, you can use an established method called canonical correlation analysis, and that allows for little relationships between different multivariate data sets. And the multiple aspect is we want to have genes and microRNAs, but we also want to have the genes and the microRNAs associated with the phenotypes. So we kind of add that into the mix. The next aspect is we may have tens of thousands, 20,000 genes at most, and maybe 2,000 microRNAs at most in human, but we don't expect all of them to be involved in the network. So we want to constrain the numbers of features and connections in the network, and you can build in some sparsity. So there's an objective function that fit, and what you get in the end is canonical weights that can then be used to construct networks. So just to show this in a more multi-omics sort of pipeline, here we have metabolized proteins and messenger RNA. We have a trait here. We have a spirometry trait, and ultimately we want to get these networks. Because of this, I mentioned the sensitivity of the methods to parameters sort of in the algorithm. We build in some methods for reproducibility, so some cross-validation, some subsampling, and in the end, you get a similarity metrics between all the features. And then that can be used with some clustering approach to create the network. So I don't have time to get into all the details, but I just wanted to show you the pipeline. So going back to that motivating data set, we had a relatively small number of subjects, 27 COPD subjects. We had microRNA and messenger RNA, and after some filtering, we got down to about 400 microRNA and 5,000 messenger RNA, and this was all in blood. And this was the network that came out to be the strongest. And you'll see in green are the genes, symbols from the messenger RNA, the gene symbols, and in purple are the microRNA identifiers. And then you have positive associations or negative denoted by red or gray. So what was not going into the algorithm is that there's this hub microRNA, and we know that microRNAs are can be master regulators, regulating many genes. That was actually not a part in the algorithm, but it did in the end find this kind of hub-like structure. So this particular network, we see genes that are related to cadherin pathway and Wnt signaling that have been already identified as an important role in COPD development and progression. And then this hub microRNA is relatively more newly annotated. It didn't have a direct link to COPD, but other lung disease like TB. However, it was highly associated with many other microRNAs that have been studied, both in lung tissue and this lavage fluid that I mentioned in COPD patients, and has been increased, and in addition to other ones that have been associated with COPD in blood. And there are microRNA families that either have similar sort of regulation patterns or similar sequences. So both we were able to find some genes in microRNAs have been already studied, but also some new candidates. Now, this was a kind of a proof of principle. We were kind of testing the method. We then were able to use it on a larger data set. So then we got excited. We got a metabolon data set. We started to work with the metabolon team and able to do this on a large scale. So on over 1,000 COPD subjects, we were able to obtain metabolomics profiles through Metabolon, and also protein mix profiles through the SomaScan profile platform. We adjusted for cell types because cell type composition can affect the levels of proteins and metabolites. And after that, we ran the SMCC network. It was a rotation student in the group, and found some networks of interest. And this is for the spirometry measurement, FEV1 % predicted. So orange are metabolites, turquoise are proteins, and then negative or positive associations are gray or purple, respectively. So what we find here are some interesting candidates. This metabolite here is a surfactant, and in its role in the lung, it is known to reduce surface tension in the alveoli in the lung. While other proteins, I'm just highlighting a few of these, is troponin. So actually troponin there's a lot of literature related to heart disease. However, heart disease is a comorbidity with COPD, and troponin is also shown to have raised levels during COPD exacerbations. So we just showed more of the link and not thinking about just one protein or metabolite at a time. And that's what this network approach provides, is that we can start to think about interactions and these potential associations that would not be evident if we just did a single protein, single metabolite analysis, but started to integrate and use these networks as a way to represent the associations. So in the interest of time, can't get into a lot of the other ones, but there are some other interesting candidates that were found. So now I'll move on to the second part of my talk, which is kind of flipping the problem and not thinking so much about the features themselves and maybe how they're interact and are associated in potential mechanisms, but thinking more at the subject level. And as I mentioned, defining new subtypes. So just a conceptual figure of what we're sort of the problem. We have this heterogeneous disease, and we can use clinical variables to think about subtypes of the disease, whether it's more emphysema or more airflow limitation, more chronic bronchitis, but we can also look at these omics profiles and see if in their sort of molecular profiles are these subjects different, and how does that relate to the clinical subtyping? So we now had, at this point, this has been an evolution. So I've been working on this project for 15 years. So we, over the years, generate more and more data, and also linking with like the TopMed consortium, we were able to generate more data. So now we had large numbers of several omics. We had the proteomic and metabolomic that I mentioned before, and over 1,000 subjects. And actually, this has now gone up through the TopMed. We now have closer to 4,000 and 5,000. And then we also had transcriptomic on 2,600 subjects. And there were about 500 that were subjects that had all three. So we've tried to do a little filtering because of the high dimensional nature. The clustering algorithms that are used for subtyping do not handle this large of sort of data sets that are in the more than 20,000 features. So we did some filtering first just based on variability and if there's features that just don't change across the subjects. We did that, but it turned out not to work so well. We were very good at finding two clusters, sex, basically sex. So we had we could discriminate the males and females because there are very strong signatures for males versus females in these omics profiles. So this comes to the issues of covariates and confounding. And we don't need to do a very sophisticated omics profiling and sophisticated algorithms to tell us who are male and female, because that's in the clinical data. So we went back to the drawing board and thought about, well, we don't want to remove those kinds of features necessarily because sex is related and age and smoking are related to disease, but we wanted to make sure we included features that were associated with the other clinical variables. So we used the clinical data in an agnostic way and just filtered it to features that had some association that was not only sex and age and smoking. And that turned out to be able to pull out some other sort of disease signals. We then used a deep learning approach called variational autoencoders to do dimension reduction, because even though we filtered down to about 2,000 features, this was still too much. These variational autoencoders can be used to create embeddings in a lower dimensional space, so about 8 to 10. And then we used a clustering algorithm for the subtyping that can cluster in subspaces of even still this 10-dimensional space. Those subtypes were then evaluated with clinical variables and also feature selection. I don't have time to go to the feature selection, but I'll talk about the clinical variables. And this was performed by actually a former MPH student in our program, who's now a PhD student, Lucas Gillenwater. So in this, we first wanted to look at each omic separately, as kind of as a baseline. So I'll show you that first. Each of the cases, each of the omics identified two clusters, and there tend to be one larger cluster and a smaller cluster. So then we looked at the clinical variables that were different between these two clusters for each of the omics. And this list is, it's small to read, but I'll give you some summaries. The transcriptomic, in addition to age, there was association with the need for oxygen therapy, for airway wall thickness, for red blood cell counts. The proteomic also had differences in age, the proteomic clusters, but also kidney disease and blood pressure and duration of smoking. And the metabolomic, in addition to age, we saw also kidney disease and blood pressure, but also other comorbidities like coronary heart disease and diabetes. So what this showed us is that these omics data sets may be in some way orthogonal. They are telling us something different. And that may be just characteristic of what the platform can measure and how that relates to disease, where maybe we're seeing diabetes show up because of maybe more related, metabolic processes using the metabolomic profile. The proteomic profile has been at the time that platform did have a lot of proteins related to heart disease. And so we were picking that up as well. So this was interesting, and I think it's very important. I think that's a learning lesson, is to understand each of your omics data sets separately. So then we decided to combine now that we understood each of them separately, we went back to our pipeline, combined them, and ran our method, basically concatenating the data. And to our disappointment, we really didn't find anything strong. We had these 500 subjects that had all omics profiles, and the clusters were just not separating well. And we understand that this disease is a spectrum, but compared to the single omics, they were just very weak clusters. We weren't expecting that. We had to, again, go back to the drawing board and thought about, in each of, maybe looking at these individual subtyping results and thinking about the subjects that overlap. What are the subjects that always appear in the big cluster versus the small cluster? So we did more of an integration post-clustering. So still a multi-omics integration, but we want to kind of look at the outliers, or the sort of border cases. So there were, as I mentioned, about 500 subjects that had all three, and about 300 were always in the big cluster for each of the omics. But then we looked at the subjects that maybe were different. These 71 were in the big cluster for the protein subtype and the metabolite subtype, but not the RNA. And similarly for the other ones, these 17 subjects were similar in the protein and the RNA, but not the metabolomics. So we looked at these three in red, the ones that, again, like I said, border cases. And that's when we started to see differences in the clinical variables. Here I have the association results. They were different on sort of exercise measurements like distance walked, but also severity of GOLD, like GOLD stage and this baseline. And here you can see this graphic that shows this GOLD stage. So 0 is at least, you go from least severe to most severe, and you can definitely see these subtypes vary by the severity, where the individuals that were different in their metabolomics profile had more severe COPD compared to the ones that may be different based on the RNA. So, it was an interesting twist. We weren't expecting kind of where this project was going, but we were still able to get insight and actually think more about the border cases and these subjects that maybe just are different. So I'm running out of time, so just want to wrap things up. I hopefully alluded to some of the learning lessons that we had along the way, but I think it's very critical to have a multidisciplinary team. We were a team of not just informaticists like myself, but we had a team of geneticists and genetic epidemiologists, of course, the pulmonologist team, and also computer scientists. And it's important to be able to work together in a multi and interdisciplinary way. Another aspect is computing needs. We didn't anticipate this, but as our data sets got bigger and bigger, we had 1,000 subjects or more. It's important to think about what computing resources you have, and also do you need, especially if you're working in the context of a large consortium, there's only certain permissions, and you have to maybe use cloud-based systems and so on. So that's planning for that is important. I did talk a little about this, but it's important to study each of the data sets separately. You do get an insight into each of them before you try to integrate them. And another common theme was data reduction. We often filter by taking out features that have low variation, but it's also important to think about some of these other dimension reduction methods at later stages in the pipeline. Sensitivity to parameters, we were building in cross-validation subsampling methods that, because results can vary drastically, there's lots of steps in an analysis, and there's some tuning parameters along the way. I mentioned the network framework that we have used, and it provides a nice visualization and also a way to interpret in associations. The covariate adjustment. In the last project, I discussed how if we didn't account for the fact that, we had sex was a very much driving factor related to a lot of these omics profiles, and we had to think about sex and smoking and age. And that is actually a lot of our thinking is how do we deal with covariate adjustment in what stage in the analysis? And finally, replication. I didn't get a chance to talk about those results here because we were one of the first at the time to have a multi-omics data set like this, so we couldn't do any replication, but now there's more opportunities through TopMed. So just wrapping up, here's my website and a QR code if you want to grab it now. And this is an older picture from our pandemic days, but I want to thank all the individuals at Anschutz Medical Campus where I work, the University of Colorado, Denver, where the computer science team is, National Jewish Health, where the pulmonary team is, and in addition to the Channing Division of Network Medicine in Boston, in our funding sources. Thank you.
Wonderful. Thank you so much, Dr. Kechris. That was amazing. I really like how you summarized it all at the end of all of the different things that you learned along the way, because I think it's very easy to lose sight on the multitude of, factors that go into analyzing these large data sets. And it really, you have to think outside the box a lot of the time. And so to get us started with some Q&A, I wanted to ask you a question on how has your research always been multi-omics centered, or did you arrive at this integrated omics model? How did you ultimately arrive at the integrated omics model for your research?
It's been an evolution. I mean, I remember I was a student, so this is dating me, when microarrays first came out. We saw our first sort of microarray experiment, and we had this high-dimensional data. I think at the time, I don't think we were even thinking about multi-omics. It's just, how do we even analyze this one platform when we have thousands of measurements? So this has evolved as technologies like yours have gone developed and gotten more popular, and we're able to do this now at a large scale with these large consortium where we can get thousands of subjects. So it's really been an evolution in the last 15 years. But definitely, even now, when we have multi-omics data on the same subjects, we always start with the individual omics analysis, and then we expand out, because you really need to understand all the nuances with each data set.
Wonderful. And with that said, I have a question that's actually for both you and for Danny, if he's able to come back online. We'll welcome Dr. Panyard back. And so a question for both of you is, if you have any, I think this might be important for the audience as well, if you have specific bioinformatics tools or resources that are your go-tos, or if it really depends on the project, if you can share your thoughts and advice for folks in the audience on that.
Yeah, well, I can start. So there's one I really like. So, again, a lot of my work initially was looking at genetic and metabolite associations. And I think we've got this linked in the chat, but of course there's GWAS catalog, which provides some great interfaces for just searching for a given metabolite and trying to find which studies have been conducted for that metabolite with genetics. There's also a website called Metabolomics, a little bit hard to find, I think, if you just Google search it, but the link I think is in the chat, or the resources. And there it's Dr. Asuri, I believe, kind of curates a list of the main metabolomic GWAS that have been published, along with very high level characteristics. So things like the population and the sample type that was used. It's great if you want to understand very quickly and find studies related to a particular sample type of metabolomic data and find out how genetics are regulating those omics. Very, very useful resource.
Yes, so good question. I think the answer is it depends. There's some really great, well-established, tested, and maintained software packages used by the community. And those kind of are the ones that have kind of withstood the test of time. I think one of the more user-friendly ones for metabolomics is MetaboAnalyst. It's a whole suite of methods. But if you're looking for more, if you're familiar with, there's Metabolomics Workbench is a database of metabolomics data sets. We often deposit there. They have a catalog that we helped develop called Metabolomics Software Catalog that has several hundred and tries to describe kind of their functionality and what operating system they work on, and what, are they R or Python and so on. So if you go to Metabolomics Workbench, and I can provide the link later as well, that's a place to start as well.
Wonderful. Those will be very helpful resources for myself and our audience. So I do have a follow-up question for Dr. Panyard. What would you approach differently if you were to redesign your study? And are there any lessons or pitfalls that you would avoid in the future?
Yeah, that's a good question. I think one of the things I would like to try, and we don't really have, I guess, time to do it, would be to try building our predictive models and looking at the GWAS of metabolites in different subsets of our data. I mentioned before that the context of your data set really matters. And I believe, what we use for the models and our GWAS were just the controls from our population, understanding how genetics were affecting metabolite levels in some individuals. Looking at those who just have disease, looking at the entire population, I think can give you some different insights. So I think that'd be one thing that'd be interesting to look into. In terms of like pitfalls or lessons learned from the analysis, honestly, it's the details. That's a challenge. So when you think about, especially when you're integrating multiple genetic data sets together with metabolite data, making sure that all the genetic data sets are aligned properly, that you've got alleles mapped the same way, that the genome versions are the same, all that stuff. It's real tricky when you're actually trying to implement the stuff in practice. It's easy to miss some things there. So just be real careful when you're integrating those data sets, make sure that they're all aligned properly. And then, yeah, I think that would be one of the biggest challenges.
Wonderful, thank you. And Dr. Kechris, do you have anything to add there?
I think I may have mentioned all the kind of the lessons we learned the hard way during my talk. But, sometimes we were very naive. We thought, we'll just throw everything together. It's like, no, that didn't work so well. Other issues like harmonization, mapping the different data sets to each other is actually not as trivial. It takes more time than it looks. And also, I think early on, we didn't plan ahead because we were kind of, you get funding, you start generating data, but we didn't always have all the different omics on the same subjects. So I think now it's more principled, and we plan for that. And of course, we have support from, funding like the consortia like TopMed to do that. So planning, I think, is an important theme too. And I think the other one I mentioned a little too is there are a lot of steps to the analysis and a lot of tuning parameters along the way. And there's an art form and some of it's experience, but it's also important to be systematic and try different values and see how robust your results are, and try to build some robustness into your pipeline.
Yeah, absolutely. So I think we just have one last question that again is a bit for both of you. And I think in your presentation, Dr. Panyard, you included a slide that had a measure of different predictive scores. So I know that one that folks use a lot are like polygenic risk scores to assess different levels of disease risk in populations. And then I think that's been translated somewhat to even individuals. So I wanted to see if either of you had comments or thoughts or opinions on those scores and how you think incorporating metabolomics data impacts the usability of those scores.
Mm-hmm. Well, I can I can start. So first, to clarify, when I mentioned the PRS models, I should have clarified that we're building polygenic models for the metabolites. So a little bit different from typical PRS, which are built to measure the risk for a disease or some condition. But that is a really good question, though, of when you've got polygenic risk scores for disease, how do you leverage metabolomics to work with those? And I can think of two different ways, and it's going to depend on the application and your goal for the PRS. So in the first case, let's say you've got a polygenic risk score that does a great job of capturing risk for disease. The next question might be, maybe not all of the genes participating in that risk score have a known function or impact on your disease. Or maybe you want other ways to track the progression of disease, over time. That's where metabolomics might be useful, where you say, all right, for a given polygenic risk score model, which metabolites are associated with the level of polygenic risk that an individual has. So that can help you kind of tease apart a little bit of the biology of what's happening in between genetics and disease. The other case might be a situation where, say, you have a polygenic risk score and it does an okay job of predicting your disease risk. And you want to ask the question, how do we improve the overall ability to predict who's going to get disease? In that case, you could think of metabolomics as kind of an additional predictor set. We have your polygenic risk score plus other measures. Of course, some people do this already with clinical variables or demographic information, but certainly metabolomics, especially since a lot of them may not be well predicted by genetics anyway. So you can think of metabolomics as maybe capturing some environmental influence that you're not going to get with any polygenic score. So the metabolites might be kind of a useful additional predictor set in your overall goal to predict disease. So I think that's kind of two main ways I think you might want to use those. But certainly it's a bit of a challenging task to integrate all that information together and to make it interpretable at the end of the day.
Yeah, wonderful. Dr. Kechris, any additional comments?
No, I think maybe that was more specific for Dr. Panyard's talk, but happy
Sure.
to answer other questions.
Okay, I think we have answered all of the questions in the chat, so we can give folks a few minutes of their time back today. But I just wanted to thank you both again. They were very insightful and illuminating talks on these incredibly large, complex data sets and how to really combine them all to answer your research questions, generate more research questions, and also providing lots of helpful insights into different resources available and how you really tackled these challenges of large omics data sets. So thank you both again. I also wanted to remind everyone in our audience that